Let's watch some videos, no, while waiting for others to join. Let's see. This one is on spectrophotometry. Intro to spectrophotometer. Okay, so our lesson for today is about quality assessment methods. So we will be talking about sensory evaluation and some of the methods used to assess the quality of fish and fishery products. So, but before that, I want you to watch these 3 videos, which are actually, links actually were given in your PowerPoint slides, no? But I think wala nyo pa ni ma-view, so though medyo mahaba sila, we will not watch the full video. So part of it only, no? Malatawan na natin ang principle because it's important para later tuloy-tuloy yung ating discussion on the methods. Okay? So please let me know if makita nyo ang video sa inyo screen. So this one is about spectrophotometer. Okay?
The spectrophotometer is a ubiquitously used instrument in biological, chemical, clinical, and environmental research. Spectrophotometry is the quantitative measurement of how much a chemical substance absorbs light by passing a beam of light through the sample using a spectrophotometer. By measuring the intensity of light detected, this method can be used to determine the concentration of solute in the sample. The beam of light that is radiated towards the sample is made up of a stream of photons. When photons encounter molecules in the sample, the molecules may absorb some of them, reducing the number of photons in the beam of light and decreasing the intensity of the detected signal. Transmittance is the fraction of light that passes through the sample and is defined as the intensity of light passing through the sample over the intensity of incident light. Absorbance is the inverse logarithm of transmittance and is the quantity your spectrophotometer will measure.
Okay, I guess we have to stop here, no? So, are you still taking, or when did you take, when did you take Fish 154? This sem or last year? Nag-take ka mo last year? Hello?
Yes, ma'am.
Last year, no? So, di ba, I hope you were able to use a spectrophotometer during your 154 or in your other courses. Di ba? I hope you have used a spectrophotometer. So, what are we reading actually is the absorbance, no? And as explained in this video, no, I don't know if na-mention niya, but as the concentration, can you see my cursor? As the concentration of your solute here increases, the absorbance reading also increases. Now, because what you are reading actually is the logarithm of the transmittance. No, so mas maliit yung transmittance, but the log of that is the one that's being recorded in the spectrophotometer. So pag halimbawa, if you're reading a bacterial culture, for example, the more concentrated the bacterial cells in your sample is, the higher would be the absorbance reading. The same would be sa mga fishery products na samples natin. If, for example, we want to read certain degradation products, no, present in our sample, if you want to check if ano na yung level, no, we can also use a spectrophotometer, and we can prepare a standard, no, wherein we can compare whether the concentration of that particular compound or substances in our sample is already an indication of spoilage, or if it is still okay for consumption or whatever. So spectrophotometer is very useful in evaluating the freshness of your product. Okay, also in fishery products. Okay, so let me end sharing this screen and let me share another one. Okay, sorry. So here's another one. This one is, unahin pa natin itong HPLC. Sige, unahin natin itong HPLC. This is HPLC. Bakit wala siyang sound? Ay, sorry. Wala nga naman pala siyang sound. Okay, ulit. Lagi na lang umulit.
Performance liquid chromatography, HPLC, is a highly versatile technique that separates components of a liquid mixture based on their different interactions with a stationary fluids. HPLC is an adaptation of column chromatography. In column chromatography, a column is packed with microscale beads called stationary phase. The stationary phase beads are functionalized for chemical groups that induce an interaction between the bead and the components of a mixture located within it. As the mixture flows through the column, the components interact with stationary phase differently. In HPLC, column chromatography is performed at a higher flow rate and therefore higher pressure than classical column chromatography. This enables the use of smaller stationary phase beads with a greater surface area toleration, which greatly increases the interaction with the stationary phase and components in the molar phase. In this video, we'll introduce the basics of the operation of HPLC by demonstrating the separation.
So you inject the sample. Ayan.
There are two types of HPLC in multi-temperature. HPLC is introduced to identical to a small volume. Sample example discrete. HPLC, the instrument is used to purify the mixture, and the desired amount of each component is collected in fractions. The HPLC instrumentation consists of a series of simple components. First, the mobile phase, solid phase, assisted by one or more elements at a constant rate. The sample is injected into the mobile phase stream by the sample injector. The sample, diluted by the mobile phase, is then to the HPLC column where the components of the sample are separated.
So ito na yung column kanina na pinakita.
And either saved in fractions for later use or transferred to a waste column. The HPLC column is the key component to a system. It's composed of a metal or plastic cylinder, not scale into stationary fluids or technical plastic. The sample mixture flows interaction with stationary fluids as it flows by. Stationary fluids differently. Travel down the length of the column.
Okay, so this one, this technique is used to separate the components of a sample. So if, for example, your sample is a protein extract, no, and you want to analyze the components, the specific components of your mixture or sample, you can use HPLC, no? So the separation principle of HPLC is based basically on the distribution of the analyte or the sample between a mobile phase, yun yung liquid, and the stationary phase, ito yon, no, yung column. So depending on the chemical structure of your sample, the molecules are retarded while passing the stationary phase. So different components will travel on a different rate. So yan yung separation principle ng HPLC. So we will not finish this video, but if you want to watch the full video, I have given the link doon sa PowerPoint slides natin. Okay, the last video. Ayan, kasi ito yung mga pag-uusapan natin mamaya. So I don't have to explain anymore, no? Later.
Gas chromatography, or GC, is a technique that is used to separate, detect, and quantify small volatile compounds in the gas phase. In GC, liquid samples are vaporized, then carried by an inert gas through a long, thin column. Analytes are separated based on their chemical affinity with a coating on the inside of the column. Because GC requires analytes that are vaporized to the gas phase, the instrument is ideally suited for volatile, nonpolar chemicals less than 1,000 Daltons in mass. For larger, aqueous, or polar molecules that are difficult to vaporize, liquid chromatography is a useful alternative. This video will introduce the basics of gas chromatography and illustrate the steps required to analyze the chemical species in a non-aqueous mixture using a sample gas chromatograph. The GC instrument has 5 essential components. First, an injection port is used to introduce the sample.
So dito mo ini-inject yung sample.
Next, a heating chamber vaporizes the sample and mixes it with an inert gas. The inert gas, such as helium or nitrogen, carries the vaporized sample through the system. Combined,
Kaya siya tinawag na gas,
sample
chrome.
makes up the mobile phase.
Ayan.
Next, the mobile phase enters the heated column, separating the analytes as they flow through.
Ayan. So dito sineseparate yung analytes or yung sample. Ayan. So meron siyang chromatogram dito. Oo.
The column is a capillary with a stationary phase matrix coating the inner walls. Alternatively, columns can be packed with matrix-coated beads. The stationary phase is usually modified polydimethylsiloxane, which is ideal for resolving nonpolar molecules. Its separation properties are refined by adding 5 to 10% phenyl, cyanopropyl, or trifluoropropyl groups. Analytes with low chemical affinity for the stationary phase move quickly through the column, while molecules.
So yung may mga molecules na may low affinity sa column, sila yung mauuna. So nagdi-differ din yung migration or yung separation ng samples. Depende dun sa sample mo. And it gives a chromatogram.
And records gases as they elute.
So dito sa sa gas chrom or sa GC, may ma-generate ka na chromatogram. So again, if you want to watch the full video, no, you can access it using the link given in the PowerPoint slide. So any question doon sa tatlo? Yun lang yung na-include ko na video, but madaming techniques yung pag-uusapan natin ngayon. Actually, I am tempted not to give a lecture on this anymore kasi binigay ko na to sa inyo na tawag dito na parang asynchronous na assignment ninyo ito. But anyway, sige, let's discuss this na lang kasi ngayon lang kayo magte-take ng Fisheries 160. Ay, teka. This is a wrong slide. So, according to the latest announcement that I have received, the last day of classes is on June 11, right? So, that's 2 Fridays from now. So, ang Fisheries 160 naman is, F160 has 6 modules. So, after this, we are left with 2 modules. So, bawat module na yon, module 5 and module 6, each module has 3 PowerPoint lectures. Ayan. So, I want you to check ahead the lectures. Kasi maiikli lang naman, nasa 16 to 18 slides lang per lecture noon. I-browse na ninyo agad para next Friday, we will meet again synchronously and we will just go over the slides. Yung mga kailangan lang i-discuss further. So, kung pwede, as much as possible, you reserve already your questions or you take note already what are the things that you wanna ask, no? Para yung dalawang modules i-discuss natin in one meeting. Okay, and then we'll have the synthesis on June 11, and we will have your exam on the scheduled date, kung ano man yung schedule ng exam. Or baka gusto nyo ba mag-exam tayo ahead sa Fisheries 160 para hindi siya sumabay dun sa maraming exam ninyo? Or gusto nyo dun na lang sa schedule? Dun na lang tayo sa schedule kasi para mabitin din, ano? Okay. Let me check ulit. Kasi meron akong klase na I thought na record ko siya, pero natapos na yung, ay hindi, dito ata yun. Hindi ko pala na-record. Yung nag-answer tayo nag-discuss tayo ng answers sa quiz. No? Item number 20 na, nang ma-realize ko na hindi siya na-record. Okay, so ganun. Kaya gumawa ako ng another video. Another lecture. Okay? So, again, we will be talking about quality assessment methods. So when you say quality, it refers to the aesthetic appearance, no, and the degree of spoilage, safety of the product. So most often, quality refers to the aesthetic appearance. Diba? Agree naman kayo doon bilang mga consumers. And freshness or the degree of spoilage which your fish or product has undergone. And it may also involve the safety aspects, right? Such as being free from harmful bacteria, parasites, or chemicals like the histamine. It is important to remember that quality implies different things to different people. And it is a term which must be defined in association with an individual product or type. Ayan, kasi iba-iba din yung description ng quality, depende din sa product. Alright? So for example, it is often thought that the best quality is found in fish which are consumed within the first few hours post-mortem. However, very fresh fish which are in rigor mortis are difficult to fillet and skin. Agree ba kayo doon? After our module 1, 2, and 3 na discussion. Agree? I believe so. Okay? And are often unsuitable for smoking. Mahirap kasi siyang i-fillet. Thus, for the processor, slightly older fish which have passed through the rigor mortis process or stage are more desirable. Kaya nag-share ako last time that when we do experiments on bangus deboning, maraming lamok dito sa office ko, bangus deboning, we prefer the fish or sample that has passed through rigor mortis or nasa resolution of rigor stage na siya. Kasi mas madaling bunutin yung bones, no? Yung bones pa naman ng milkfish, madaling maputol. So talagang sinasadya minsan na medyo ipapass through muna siya ng rigor mortis before siya i-debone. Okay? So since the consumer is the ultimate judge, no? Ito naman ang safety should be free from contaminants, handling, preservation, and natabunan yung ating and storing of food in ways to avoid, limit, or reduce the risk of foodborne diseases. So most chemical or instrumental methods must be correlated with sensory evaluation before being used in the laboratory. So ganun naman talaga. The sensory method or analysis is used side by side with your chemical and microbiological tests. Okay? Ayan, it should be performed, yung sensory analysis should be performed scientifically under carefully controlled conditions so that the effects of test environment, personal biases of the panelists, kasi tao yung nagtitest, no? And others may be reduced. So medyo hindi siya ganun kadaling gawin yung sensory analysis. Akala natin, kakain lang tayo ng sample and then magre-rate kung ano yung masarap, alin doon yung gusto natin, but it's not that easy as we think. Ayan. Ayan. So, what are the methods to assess the quality and safety of fish and fishery products? So we have I categorized it into three. We have the sensory evaluation, the microbiological testing methods, and the chemical or instrumental methods. Yan. So, first off, yung sensory evaluation method. So, we have a definition here, no? So, basahin nyo na lang. Most sensory characteristics can only be measured meaningfully by humans. Iba yung judgment ng human. Ayan. So, however, advances are being made in the development of instruments that can measure individual quality changes. So, yung texture, meron na tayong texture meter. Yung color, meron na rin tayong colorimeter. Ayan. Kasi yung instruments kasi, it eliminates the biases that the human panelists may have, no? Instruments are capable of measuring parameters included in the sensory profile, and examples of those are the instron, the rheometer for measuring texture and other rheological properties, lalo na yung mga surimi natin. Microscopic methods combined with image analysis are used also to assess the structural changes in the artificial nose. Merong artificial nose to evaluate the odor profile. So, unti-unti, nagkakaroon tayo ng hindi naman siya replacement but alternative to human panelists. Ayan. So, let's see if I have here a process on how to do sensory evaluation. Actually, wala pala siya. So, I will discuss na lang. So, sensory evaluation, the conduct of sensory analysis is actually has or requires several requirements. May mga requirements yon. First off, yung sensory analysis room or the room that you need to use for your sensory evaluation should be well lighted. Yung wall should be, yung color ng wall should be off-white or white. And yun, sufficient dapat yung light. Ano yung reason? Bakit ganun dapat yung sensory room natin? Bakit off-white? Hindi pwedeng red, hindi pwedeng blue, orange, ganun. Bakit dapat white yung ating wall sa sensory room? Anybody? Okay, the reason for that is para hindi maapektuhan ng color ng wall, yung color ng ating product. Especially if color is one of the attributes that we are looking at or we are evaluating. Ayun, so ganun dapat. Hindi siya dapat maka-affect doon sa judgment natin. Also, there should be a separate room for product preparation and the sensory evaluation. Room, kumbaga, yung room kung saan yung panelist mo nakaupo at magte-test ng product should be separate from your room na ginagamit pag nagsaslice ka, naglalagay ka sa plate, gano'n. Hindi dapat makita ng panelist mo. Panelists are the ones testing the product. Okay? Hindi dapat makita ng panelist mo yung samples before niya i-test. So, dapat makita niya pag mag-a-analyze na lang siya. Kapag hindi pa siya mag-a-analyze, piniprepare mo, hindi niya dapat makita yon kasi hindi rin pwede na may idea siya kung ano yung mga nilalagay mo na codes, no? The panelist should not be aware na this code, an example, 555 is the one that has a lower salt na dried fish. Yung 999 has higher salt. Ganun. Okay, when you code your product or your sample, you use a three-digit random codes. So numbers yon, 3 digits. So halimbawa, 153 or 369, mga ganun. Walang meaning yon. Okay? So as to avoid bias. Kasi pag sinabi mong sample A, sample B, sample C. So parang ma-associate mo siya. Maybe A has this level of saltiness and B has a bit higher saltiness. C is higher than B. Parang ganun, ma-associate mo siyang ganun. But if you're using 3-digit random codes, that eliminates that particular bias. No? Another thing, if odor is one of the attributes that you are trying to check in your sample or in your product, you have to prepare your product na naka-foil, no? So that pwedeng i-open nung panelist mo yung product na maaamoy pa talaga niya kasi na-preserve mo yung odor doon sa inside your product. Ayun. What are the other requirements? Ayan. Ano ba yung requirements? Dapat when you conduct your sensory evaluation test, your panelist is neither gutom nor busog. Ayan. Ano naman yung reason? Bakit hindi pwedeng gutom na gutom si panelist o hindi din pwedeng busog na busog siya? Actually, mas hindi pwedeng gutom siya. Pwedeng okay na lang na busog siya, pero hindi pwedeng gutom siya. Okay, the reason for that is everything na ipapatikim mo or everything that your panelists will taste will actually taste good and masarap, lalo na pag gutom yung tao. Okay? At some sort, medyo hindi objective yung magiging judgment ni panelist kapag gutom siya. Okay, another requirement, kailangan meron siyang source of water na halimbawa you are testing or evaluating 5 products. So every time you are done evaluating, every time you evaluate a product, after mo nun, you have to rinse your mouth. Kasi you will be tasting another product. Iba naman yung concentration ng ingredients nun or iba naman yung qualities nun. So you can, parang nag-start ka from zero. So you have to rinse your mouth, you clean your mouth, then dapat merong sink doon mismo sa evaluation room, no, where the panelist can, yun, magmugmug siya. Alright? And sometimes you are giving your panelist nakalimutan ko yung tawag sa rice, para maging bland ulit yung kanyang panlasa. Ayun. So, merong ganon na requirements. And sometimes, meron talagang booth for sensory evaluation. That's called a sensory booth. And sometimes, evaluation is done on a round table type. So, pwedeng magkaharap yung lahat ng panelists and then they do the evaluation of the product. Okay, medyo natagalan ako doon, no? Let's proceed sa next na slide natin. So, this one shows the types of sensory testing. So, we have the difference test. No, of course, as the name implies, you are trying to identify which of the samples given to you is different from the rest. Ayan. I will give a specific example in the next slide. Okay, next is the rating test. Okay, so in the rating test, no, you can do ranking, single sample test, 2 sample difference test. O yun, i-rate mo lang yung product. Halimbawa, saltiness, from 0 to 10. Ano yung level of saltiness nito? Ayan. So, ganun yung rating test. Next is the descriptive test. So, you describe the product, no? And the last one is the sensitivity test. So, you determine the sensitivity threshold test using dilution test. Ayan. So, yung hedonic rating test, yung hedonic, ito, ayan, you are given a scale, no, and then you rate kung ano yung score ng product, depende dun sa na-perceive mo. Yung numerical scoring test naman, parang hedonic siya, pero may number. So, from 1 to 9 or 1 to 10, you rate. Ano ba yung score mo sa kanyang color? In terms of whiteness, ano siya? 10 ba siya? 5, 1, 0, ganon. And composite scoring naman, composite scoring test, rating is defined so that specific characteristics of products are rated separately. So, for example, when you say test or rate the itchiness of the product. So, when you rate the itchiness, di ba? Usually, pag 10, yun yung very itchy. Pag 0, not detectable. Pag 5, moderately detectable, ganon. But pag sinabi mo naman na rate the flavor of the product. So pag sinabi mong 10, di ba, yung 10 is the most positive. So, most desirable. Number 5, neither desirable Or yun, ano yung negative nun. And then yung 0, dislike extremely. Parang dislike extremely, neither like or dislike yung 5. Yung 10 mo is like extremely. Ganon yung composite scoring. So for iba yung pagka-define mo ng score sa histamine or itchiness, iba yung pagka-define mo sa flavor, iba pa yung pagka-define mo sa odor, mga ganon. Iba yung definition. So that's for composite scoring. Okay? So this is the example of ano ito kanina? Difference test. So we have the triangle test, no? So which of the itong strawberries natin, which of the samples is different? So i-test mo lang sila and then you have to spot the difference. No, the spot which one is different. Okay? And then taste, ito namang do trio test. You have a reference sample represented by ours. Ano yung aking cursor? Ayan. You have a reference sample represented by this R. And then you identify which of A or B is similar to R. So that's a duo trio test. Okay? Dito naman sa paired comparison test, you have to identify between the two samples kung saan yung mas sweet or sweeter sa kanila. So, ganon. Usually, you do this if you want to compare your newly developed product versus a product in the market. Halimbawa, meron kang na-develop na fishery product or canned fish, for example, and you want to compare it with Mega Sardines or 555 or Ligo, and gusto mong tingnan kung identify ba nung panelist mo yung product mo, or does your product taste like the commercial one? O ganun yung purpose na ganitong test. Okay? Ayan, ito naman yung example ng hedonic na sensory score sheet. Okay? So, halimbawa, ilang points ito? 2, 4, 6, 8, 9. Ayan, so you will be asked, nababasa nyo ba? So, instruction. This is a sensory score evaluation for evaluation score sheet for steamed fish. In a scale of 1 to 9, please evaluate the given samples based on their attributes. So, i-rate mo, dito naka-specify na 1 is the lowest and 9 is the highest. So, in terms of appearance, bahala na kung ano yung appearance, depende kung gusto mo siya, you rate it 9. Kung medyo hindi, so towards 1 ka. So, ito yung example ng sensory scorecard. Have you ever done sensory evaluation? Nung second year, third year kayo, nakapag-sensory ba kayo? May nag-sensory na ba sa inyo? Hello? Nakapag-sensory na kayo, class? Hindi pa, wala pang naka-experience sa inyo ng sensory evaluation? Ayan. So this one shows the descriptive test, no? Ito, dinidescribe talaga. As the name implies, dinidescribe yung product. So, dito, ginigrade yung freshness of fish, no? Using the EU scale. Ayan. So, ititingnan yung, may trained assessors, tapos ititingnan yung skin, outer slime, eyes, gills, belly cavity, and then i-grade siya according to extra, grade A, and grade B, ganon. So, of course, the extra fresh is much fresher versus A, and A is fresher than B. Ganon yung rating ng EU scheme. However, ayan, EU scheme takes no account of detailed species differences. So, meron kasing magkaibang species, no? Pag ibang species na yung ginamit mo, parang hindi na applicable yung ganong scheme. So, mag-iiba na naman yung parang way of evaluating the product. So, hindi siya species specific. Okay? Kasi isang ano lang yun isang scheme lang siya. Wala siya yung hindi sila nag-develop ng specific for certain species. General siya, yon yung term. Okay, it does not describe accurately different levels of freshness quality. So fish in the same grade may have quite different freshness characteristics. So ganon yon. Meron silang top A, top extra, lower extra, mga ganon. So this gives no information on the remaining storage life. So ayan, ito yung i-introduce natin, yung next na scheme. Kasi yun yung limitation ng EU scheme. This one is about the principle, the QIM or the quality index method. Ayan, so this one is appropriate sensory attributes are evaluated in sequence. So ito yung mga sensory attributes. So meron silang mga parameters na tinitingnan, the appearance, the body stiffness, odor, eyes, gills, yon. And then it has assigned mga demerit scores. Ayan, so demerit scores yan. Dito sinabi, each attribute is scored from 0 to 3, 0 indicating the best quality. Kasi naman, when you say demerit, the higher the value, the lower yung quality. Kasi demerit nga siya, negative. Ayun. So the scores of all attributes are summed to give the total demerit score. So obviously, the higher the score, the demerit score, the lower is the quality of the fish or the product. Excuse. So the quality index increases linearly with time of ice storage. So making it easy to predict the remaining shelf life. Ayan. So pero take note, a separate scheme should be developed for each species. Kasi merong mga species na for example, yung ice nila iba kapag fresh towards pastel sila, iba yung itsura. So ibahin mo din. You have to also change the scheme. Ayan. So ito yon. Sinabi kanina, quality index increases linearly with time of ice storage. So you can, using the quality index method, you can actually predict the remaining shelf life of fish stored in ice. So ito yon. Sorry guys, nag-class, may remnants pa ako ng aking ubo. Excuse me. So ayan. So 'di ba ito 'yon? Can you still remember this graph? So pinag-usapan natin 'to during our module 1 to 3 na discussion, no? So it has 4 phases. So ito yung sensory score, quality score. So ayan, pababa, 'di ba? Obviously, bumababa yung quality ng fish as the days it is stored in ice increases. So ayan, ito so upon reaching this rejection level, ito yun, no, 'di ba? So ipag igaganon mo siya, it will meet here. So meaning at score of 4 wait. I'm sorry. So at this particular quality score, mare-reject na yung product. At inabot na rin niya yung phase 4, no? And then yung demerit points niya is 8. Dito. So ayan, tingnan mo dito. So halimbawa nagkanda kayo ng sensory analysis using the QIM scheme and your score is 10, you will be able to plot it here, no? Titingnan mo kasi ito yung 20, tapos ito yung i-match mo dito na you can keep your fish for 10 days at 0 degrees Celsius, no? Kasi at that time you will be getting ayan. Ito yung halimbawa, you get a score of 10. So you will be able to determine, ipa-plot mo siya, tapos kukunin mo yung number of days remaining in ice siya. So ito yung malalaman mo. You still have 5 days para ma-store mo pa siya sa ice. So halimbawa, 0 yung demerit score mo. Pag 0 siya, you still have 10 days to keep your sample in ice. Halimbawa naman, yung score mo is 20 na. So ito yung remaining days in ice, hindi mo na siya pwedeng i-keep pa further sa ice kasi deteriorated or nag-stale na yung product. So any question with this graph? Hindi ba kayo na-confuse? So ito ha, remaining days, storage time in ice. Ito naman, days in ice. So you use this information in order to come up or to make this graph for you to be able to predict the remaining days in ice of your sample. So ganun yun. This is very useful. Ayan. So this one shows the scheme for the evaluation of cooked fish. Okay. So, tingnan nyo, the score here is 10, no? Para sa mataas na score. And then, dito naman for the odor, the highest score is 5. And for you to reject it, you have this scoring. So, what can you say about this? Para siyang composite scoring, no? Para siyang composite na na type of composite scoring test, na type of rating test, right? Do you agree? Natandaan niyo pa ba yung composite natin kanina? Ayan, meron akong quote dito. Oy, original quote to ha. Do self-evaluation and determine the products of your life. Avoid doing the same to others. Avoid evaluating others. Ayan. Ibig sabihin, gusto kong sabihin, huwag pakialaman yung iba. I don't know, bakit yan yung naisip ko na quote that time when I prepared this lecture. By the way, ako yung nag-prepare ng lectures sa module 3 and 4. Ayan. So now let's go sa microbiological testing methods. So this will be quick, right? Microbiological analysis is the use of biological, biochemical, molecular, chemical methods for detection, identification, or enumeration of microorganisms in a sample. And it is often applied to disease-causing and spoilage microorganisms or yung ating tinatawag na SSOs, 'di ba? Alam natin yung SSO. Yan. So and microbiological testing methods are used to evaluate the possible presence of organisms of public health significance. So this could help us assess the hygienic quality of the fish, whether the fish has been subjected to time temperature abuse, and also to determine the degree of microbiological contamination of the equipment used, the surfaces, the workers, during the preparation of the product, and also to establish standard of hygiene during handling and processing. So, pwedeng gamitin mo yung information mo doon sa microtest mo to establish yung mga SOPs, mga standard operating procedures in your plant or in your laboratory. Ayan. So, what is the purpose of conducting microbiological tests? One is to meet the desired specification for raw material, intermediate, and finished products. Kasi later on, you will see that for a certain product, there is a microbiological limit that's been set by the EU market, by Japan, and also by US. So if you wanna export your product, you have to comply with those microbiological limits. Okay, masyado akong mabilis magsalita. Do you have any question before I proceed? May tanong, class? Okay, I'll assume na wala kayong tanong. Basta, I hope you are following, no? So, next is to assess quality and safety of products. So, that's obvious. Identify the risk factors. Also, kasi pag alam natin na mataas yung salmonella, for example, sa product natin. So, alam natin kung ano yung potential risk, kapag mataas yung salmonella. And also, if we know that our canned product has high levels of Clostridium botulinum, we know that Clostridium botulinum can produce toxins, that can be detrimental. So, alam natin yung mga risk factors, okay? To detect the presence of foodborne pathogens and toxins, yun din yun. And then to verify the process no, in producing the product. Ano yung ibig sabihin ng process? Okay, we do microbiological tests to check whether our process is effective in eliminating the pathogens or the certain types of microorganisms that we don't want to be present in our product. So, halimbawa, you are producing a dried product. Okay, so initially you have to also test your raw material if mababa yung load ng example, gusto mong i-test yung E. coli level dun sa, sige, yung example na lang natin na gawin is yung green shell or yung tahong. Ayan, Pernaviridis. If you wanna check if yung depuration method mo is effective, do you know what a depuration process is? Yun yung, kung naalala niyo yung ginagawa ng nanay niyo, pag bumibili siya ng green shell or ng, hindi ko maalala yung name nung bagungon, yung nilalagay. Ano ba yun? Basta shell, mga shells. Pinapasuka sila, di ba? So, overnight nilalagay sila sa water para i-release nila yung, especially yung mga filter feeders, i-release nila yung dirt, no, na nasa system nila. So that that process is called depuration. So if you want to check whether your depuration process is effective in eliminating or reducing the amount of microorganisms present in your sample, pwedeng before nun, upon capture, i-test mo na yung micro level niya, and then you, after depuration for certain or for a number of hours or days, you check again. Kung bumaba yung microbial load, then yun yung ma-verify mo that your depuration process is effective. Okay? Sa cooking, sa preparation naman or processing of the product, pwede mong i-check whether after drying your fish, na-reduce mo ba yung amount of bacteria na present sa product? Yung implication noon pag na-reduce mo is possibly you have reduced the amount of microorganism because you also lowered the water activity. Okay? I don't know if you have discussed water activity in the past modules, no? But water activity is one of the requirements of microorganisms, in particular bacteria, for them to grow. So pag hindi optimum yung water activity for their growth, they will most likely not grow in your product. Or they will be eliminated or malilimit yung growth nila. Okay? So this one naman shows the parang yes or no na scheme, no? Has the food been associated with disease or does hazard analysis indicate reason for concern? So pag hindi mo naman na subject doon yung product mo, na halimbawa, hindi siya na-expose sa high temperature for several hours, no? So no need of testing. Ito yung mga guide, no, for you to know whether you need to test your product or not. But in fisheries, no, we usually test our product. Kasi kailangan natin yun. And we also do, as I've said kanina, we have to perform sensory evaluation side by side with your micro and your chemical test, no? Para malaman mo if, for example, nag-change yung attributes, sensory attributes ng product, may role ba doon yung microorganisms or does the growth of microorganisms or the increase in number of microorganisms in your product affected the flavor, the taste, no, the taste and the odor of your product? So ganon, i-correlate mo sila lahat, tatlo sila. Okay? So ayan. So ito naman, medyo microbiology ito, no? This talks about culture media. So we have different types of media, right? And culture media is composed of different nutrients to enhance the growth of microorganisms. So yung enrichment, we have enrichment media, supportive media, selective media, and the differential media. So yung difference nito is yung enrichment media, it allows the growth of a wide variety of microorganisms. No, maraming microorganisms yung pwedeng mag-grow dito sa enrichment media. Ito namang supportive media, you are adding certain nutrient, no, to allow the growth of fastidious organisms. Like for example, you add blood cells para ma-promote mo yung growth ng mga microorganisms that requires or bacteria that requires blood cells for growth. Ayan. And then for selective media, as the name implies, no, you only allow the growth of selective microorganisms. All right. So sometimes if you don't want this particular bacteria to grow in your media, you add an antibiotic for that microorganism para pag may antibiotic na ganun doon sa media mo, yung microorganism na sensitive for that antibiotic will not grow, of course. So that's an example of a selective media. And then yung differential media naman is used to grow or used to differentiate a certain or specific species from a group of related species. So for example, gusto mong i-identify yung Vibrio species. So you use a TCBS instead of simply using using a nutrient agar, kasi yung nutrient agar, nag-cater din siya ng growth ng Vibrio species. But if you use TCBS, no, that's a green media specific for Vibrio. Ang mag-grow lang doon yung mga Vibrio species. And then when they grow, makikita mo yung mga luminous na species, it will emit light kapag nilagay mo sa dark yung media na yun. So that's an example of a differential media. Ayan, so this one shows the pathogen and then the selective differential media that you can use, no, to grow these specific pathogens. And then we have the selective agents that you can add, no, in your media. And this one shows the inhibitory actions, no, of using this medium. Okay, so you just check this slide further later para tutukan nyo na lang siya mamaya. Okay, we'll discuss APC. So APC stands for aerobic plate count, and this is used as an indicator of bacterial population on a sample. So pwedeng yung microbial limit mo will have an APC of 10 times 1 times 10 raised to negative 5. Ganon yung limit, no, for an APC. So it does not differentiate types of bacteria. General siya, total count yung tinitingnan, and can be used to gauge sanitary quality, organoleptic acceptability, adherence to good manufacturing practices or GMPs, and to a lesser extent, it can be used as an indicator of safety. Kasi obviously naman, kapag mas malaki yung population ng microorganisms sa sample, parang ang indication nun is it's already unsafe for consumption. No, at some sort, we allow an APC up to 10 raised to 5. No, pag 5 yun, maximum of 900,000. Oo, 900,000. 999,000. Oo. So, pero beyond that, we already consider our product to be unsafe. Okay? And it may provide information regarding the shelf life or impending organoleptic change in a fish. Okay? Bakit natin ina-associate sa quality na kapag mataas na yung microbial level ng ating product, we also consider na there will be changes or effects dun sa organoleptic properties. Kasi, di ba, we know that microorganisms are capable of producing enzymes, and they are also capable of degrading and using the fish as their media. Kakainin nila yon, and that they produce metabolites, no, that can eventually be attacked by endogenous enzymes and also enzymes produced by the microorganisms. And yun will trigger a change in the quality, organoleptic quality or attributes of the food product or fish. Okay? Large numbers of bacteria may be an indication of a poor sanitation or problems with process control or ingredients. Ayan. So, TVC naman refers to the total viable count. Anong pinagkaiba niya dun sa APC, no? The TVC is defined as the number of bacteria, CFU, in a food product. Yung APC, aerobic plate count, this one naman refers to the viable count, yung mga buhay. Okay? The total viable count develop into clearly visible colonies. Okay, and when the test is carried out under standard conditions, TVC is only a measurement of the fraction of the microbiota able to produce colonies in the medium used under the conditions of incubation. However, meron tayong problem, no? There are microorganisms that are non-culturable. Hindi sila kayang i-culture, no? So, hindi mo sila makakount doon sa iyong TVC or APC. So, limitations of this test cannot measure the total bacterial population. Alright? Kasi yung mga hindi non-culturable cannot be included in the count. No differentiation between the different types of bacteria. So, hindi mo malalaman saan dyan yung mga enteric bacteria, saan dyan ang SSO, saan dyan ang ganitong type, no? And doubtful in the examination of, can be, kapag ito yung test na ginamit mo, will be doubtful if you use, if your sample is a frozen fish. Okay? Kasi, pag frozen yung fish mo, some of the microorganisms would also be dormant or natutulog kapag frozen. So, hindi sila ma-include dun sa mga viable counts. Okay? Kasi yung viable count mo lang naman is yung microorganisms that are capable of producing colonies dun sa media. Okay? Nag-enjoy ako class kasi parang feeling ko ang ganda ng aking background. Ayan. Ngayon ko lang siya na-discover. Hay nako. Patapos na yung SEM. Okay. So, TVC should be not, ano to? Ayan. Kapag ang TVC mo or total viable count mo is greater than 7 times 10, no? Or 7 log CFU per gram. So, ibig sabihin nun, 1.0 times 10 to the 7. Ayan. This can already give you a detectable na changes in the organoleptic properties of the product. So, yon, may mga spoilage na, and then can no longer be acceptable for consumption of product. Okay? So, the limitations of TVC, ayan, ito yung kanina, yung nasa blue na box, no, yung tatlo. Ayan, bumalik tayo sa SSO. SSOs must be enumerated and identified for quality control or determination of the remaining shelf life of fish. No? So, iba pa itong SSO. Ayan. So, these are organisms that are specific to each species of fish. Okay? So, na-define natin last time yung SSO, na those are the microorganisms that are capable of producing spoilage or producing metabolites, no, dun sa products natin. Okay. So, in addition to aerobic plate count, the following methods are useful for foodborne pathogens and toxin detection. So, we have the antibody-based method. So, meron tayong ELISA or the dipstick assay. Kasi kung gusto mong i-detect yung presence or absence of the organism, you can use a dipstick assay. Para siyang pregnancy test, no? Na mag-show siya ng line kung present yung microorganism na yon, and kapag absent naman, it will not show any line. Yun yung example ng dipstick assay. Okay? Or it will cause a change in your test, ano siya yung tawag, parang test na paper. Parang litmus, ano siya, parang pH paper na pag dinip mo doon, mag-change yung color, ganyan. So, it's an indication that this particular microorganism is present. Your SSO is present. Ayan, we also have nucleic acid-based assay, the PCR or the polymerase chain reaction, the DNA hybridization, and the DNA microarray. Or we can also use biosensors. So yung nucleic acid-based assay and the antibody-based and the biosensors are ideal for microorganisms that are non-culturable. Especially itong nucleic acid-based. Kasi DNA lang niya yung kailangan natin. Kahit patay yung microorganism, you can still detect its presence in your product. Okay? Ayan. So, this one, this very colorful slide is from the study of Zhuang et al. No, this shows the brief procedure of culture independent methods used in microbiota characterization of fish and crustaceans. So, bakit siya tinawag na culture independent? Because you don't need to culture the microorganism for you to be able to identify them. You just need their DNA, no? So for you to be able to ID, para actually mas maganda ito kasi you can identify the microorganism to species level because of the so many DNA sequences available online, no, na pwede mong i-match doon sa ma-determine mo na sequence. So how do you do this, no? First you do, can you see my cursor? First you do DNA extraction. All right, nowadays there are a lot of DNA extraction kits, no? You just use a reagent, no, for you to be able to extract the DNA of a microorganism. So actually yung PCR or polymerase chain reaction is ginagamit din natin to identify mga parenting analysis, to do parenting analysis, identification of detection of a disease, and also ito din yung ginagamit, 'di ba, sa pag-detect nung COVID-19 virus. No, pero yung variant nung PCR na ginagamit sa pag-detect ng COVID-19 virus is the RT-PCR or the reverse transcriptase. Kasi mRNA nung COVID-19 virus yung dinidetect doon. Ini-extract. Ayan. And dinidetect ng primer. Ayan. So, yun. Going back, no, so you have to extract the DNA, no? Halimbawa, meron kang product na meron kang fishery product and natubuan siya ng kung ano dyan na hindi mo kilala. So you take that sample, you put it in a tube, and then you add the DNA extraction reagent, and then you extract it. Usually when you extract it, you homogenize, and then you add the reagent, and then it's a centrifuge, you decant, ganun yung procedure. Okay? So once you have your extracted DNA, you do PCR or polymerase chain reaction, you amplify the DNA. Ibig sabihin nun, kasi maliit lang yung sample na kinuha mo, right? And then you just have to extract the DNA. So most likely, very, 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 very small amount of bacterial DNA yung na-isolate mo or na-extract. You need to do PCR. The purpose of that is to clone several copies of the DNA that you have extracted. When you say clone, kukopyahin mo, padadamihin mo yung DNA na na-extract mo from your sample. So, and that's how PCR works. And then, you can do DNA fingerprinting or you electrophoresis, and then you separate the amplicons, and then you submit I don't know kung nakagawa na kayo ng electrophoresis, but anyway, you can slice the bands here and then submit it for sequencing. Once you have your sequences, yung sequences class, ano lang yun? Sequences of nucleotides. So, pwedeng AC, GC, ganon. Ayan. We will learn that when you take genetics. Meron ba yung genetics? May genetics na kayo? Wala pa. fish genetics? Wala pa ba? Hello? Okay. So, I'll just proceed. Basta andyan lang kayo, class, ha? Okay, so after you've received your sequences, you can compare your sequences from the ones available in the NCBI, and then you will be able now to identify ano yung microorganism na na-extract mo doon sa sample mo. So that's how easy culture-independent technique is. Kaya lang, it will take time, of course, to submit your sample for analysis, and then, ayan, ano mo siya, i-identify. Pero from the PCR test alone, if you know the primer that you're using, halimbawa yung primer mo is specific lang siya for E. coli, you can use that. And then dito pa lang sa electrophoresis mo, you will already identify if E. coli is present in your sample kasi alam mo kung ano yung size, and then may corresponding size itong mga bands na yan. So you will identify kung present ba si E. coli or not, yung target organism mo. Okay? Sige. So I'm sharing with you this quote. Microorganisms can be good, bad, ugly, or useful. Just master how to deal with them properly. So just like people. Ayan. Pa-quotes tayo today. Okay, for the third method, we have the chemical and instrumental method. So ayan. The first is the pH determination. So changes in the pH of the fish mussel is a good index for freshness assessment. Bakit? Ano nga yung pag-usapan natin last time? Anong mangyayari sa pH ng fish? Bakit siya good index for freshness assessment? Anybody? Ba sabi natin post-mortem, the pH of the fish becomes from neutral, no? It drops to yung pH niya is na nagiging slightly acidic, all right, because of the accumulation of lactic acid, no, as one of the products of anaerobic glycolysis. So yun. So ito ang pH niya can also be correlated dito sa red meat, no? Yung pH niya can also be correlated with the color of the red meat. Ayan. So yan yung example natin. Ayan. So this one shows the nucleotide degradation in fresh fish, no? And this represents a problem due to the accumulation of some derivatives such as inosinic acid, inosine, and inosine ribose, and hypoxanthine. All right. So we know that immediately after death, ATP, no, and other related compounds are broken down by endogenous enzymes. Now, following the scheme represented in this slide, ayan, ATP down to ito. All right. So the flavor of fish, of marine products are best immediately after catch. But because of the rapid formation of IMP, no, yung alam natin sa IMP is responsible in the characteristic sweet flavor, meaty flavor of the fish, no? Rapid yung formation nito, and it gradually is converted or degraded into hypoxanthine. So ayan, ito yung representation. So during this stage, no, na nag-accumulate siya ng IMP, very tasty yung fish natin. Kaya lang, eventually, magde-develop na rin siya ng kanyang degradation products na hypoxanthine. So, this one gives of a bitter taste. Ayan. So, you should know the difference of IMP and the hypoxanthine. So, these are the quality index, no? Or pag sinabi mong index, yun yung mga tinetest, no, to identify the quality or to determine the quality of the product. Okay, so these are the methods. Kaya sinabi ko kanina na it's very important for us to watch the video before we start kasi we will be talking a lot of those, no, a lot about the video, about the methods no dito. So I don't need to elaborate no further this method. So if you want, you can watch again the videos, ayan, and for you to appreciate, no. But i-discuss natin pa isa-isa ito sila. All right. So for the K value is an index no used to measure the enzymatic freshness of fish and squid. So ito yon, K value equals, ito yung mga degradation products over lahat sila. So the ATP, ADP, AMP, IMP, hypoxanthine, and inosine and hypoxanthine. So, bakit ano siya? Kumbaga, tinitingnan yung ratio ng hypoxanthine versus the other nucleotides or yung mga byproducts of ATP degradation. So, if your K value is greater than 20%, the fish, your K value is less than 20%, that's an indication that your fish is still very fresh, right? If the K value is between 20 to 40%, no, the fish is already considered, is still considered fresh, no, to be consumed after cooking. Pero pag fresh mo siyang i-consume, hindi siya advisable to be consumed fresh. Okay? And then if the K value is above 40%, the fish is already inadequate or inappropriate for consumption, unsafe for consumption. Ito yung mga standards ng Japan, no? So the K value should be less than 10 for a fish that have just die, no? Kamatay lang. Very fresh siya for sashimi grade siya pag less than 20%, pag nasa 30 to 50%, ordinary, medyo stale na siya. Unacceptable na siya kapag above 65%. Ano yung nakikita niyong slide?
Yung module 4 pa rin po, ma'am.
Okay. Thank you. Teka, tingnan ko. Nasa na ba tayo? So, ito, ion exchange chromatography. Ayan. Tama, no? Dito tayo kanina. Ay, naku, istorbo ito. Teka, i-move ko lang yung ating beautiful faces dito sa kabila. Kasi hindi ko siya natatabunan niya yung ating slides. Okay. Going back, tingnan ko ulit ha, baka hindi tayo naka-record. Okay, good. Okay, so ion exchange chromatography is based on the separation of ions, no? And, okay, it's based on the separation of ions and polar molecules based on their affinity affinity to the ion exchanger. So later you will see here, no, na meron din siyang parang column. Okay? And it separates biomolecules based on their differences in their net charges. So at certain pH. So titingnan ninyo dito, meron tayong an ion exchange resin and meron tayong cation exchange resin. Ayan. So ayan. So meron siyang mobile phase and meron siyang stationary phase. So yung mobile phase niya could be a buffer, no, na may certain pH and no pH and salt concentration. So yung opposite charge solute ions attracted to the stationary phase, yun yung column, no, will be attracted to the yung opposite charge will attract doon sa stationary phase. And then yung stationary phase naman na nare-refer dito is yung resin, no, used to covalently attach anions or cations into it. So tingnan natin itong next slide, no, para mas maintindihan. Ayan, so meron tayong ganyan. So, ito yung tingnan ninyo. Ito yung cation exchange, no? Ito naman yung anion exchange chromatography. Pag sinabing cation, positive siya. So it bears a negative charge para ma-attract niya yung mga cations. So ayan, don't be confused. Yung anion naman, it bears a positive charge, no, to attract the anions. Kaya sila may pangalan na cation for cations and anion exchangers for anions. Okay? So ito, yung cation has negative charge, no, to attract the positively charged molecules. So hindi lang siya limited dun sa proteins. And then the anion has a positive anion exchange chromatography, has a positively charged column to attract the anions. Ayun. So I hope this one is clear. So pag so if you have your column, no, first you the procedure would be an impure protein solution, no, will be loaded dito sa iyong column. And then all the positively charged molecules will be attracted. Magdidikit siya dito sa iyong column. And then you add a buffer, no, para ano ba, 'di ba? Nag-pour ka. So pag pour mo, yung mga hindi positively charged, 'di ba, ma-elute sila, lalabas sila yung liquid. So yung matitira doon, yung mga molecules that are attracted doon sa column or sa exchanger mo. So, para ma-remove mo ngayon yung mga nagdikit doon na positively charged na molecules, magpapapass through ka ng buffer. So, yun yung i-collect mo dito. Ito yung mga positively charged protein molecules. Ayan, same way with your anionic or anion exchange chromatography. You will be able to collect your negatively charged proteins. So, attach muna sila dito. And then after that, you pour a buffer or a solution para ma-remove mo sila dito, and then you'll be able to collect them. So now you have separated your proteins by their charges. Yan yung purpose ng ion exchange chromatography. So for key value determination using IEC, no? So you use an anion exchanger, no? So kasi gagamitin mo yung anion to attract an anion molecule. Ayun. So dito you put your sample dito sa column and then you wash it with water, dilute with 45 ml solution A. And then ayan, so sa unang pag-elute, yung lalabas is the hypoxanthine, ayan. And then you read this. Ito yung mauunang lalabas. Alright? And then you read the absorbance of this. And then sa second solution mo, you elute also your solution B, and then you collect it. Ito yung mako-collect mo, yung ATP, ADP, AMP, and IMP. Now, you read also the absorbance reading or the optical density reading at 250 nanometers, and then you will now be able to compute for the K value. So ganon, yun yung gamit ng ion exchange chromatography to determine the K value of your product or your sample. So yan. Ay, bumabalik siya. Ayan. For this one, you need to measure the lipid oxidation in fish. So we know, diba, that lipid oxidation involves 3 processes. So we have the initiation. I don't know if meron yan dito. Ayan. And then initiation, the propagation, and then the termination stage. So during the early stages of lipid oxidation, we can use PV test or peroxide value test, no, to determine the primary oxidation products. Ayan, dito yon. And then to determine the secondary oxidation products, we use the T-bars assay or the thiobarbituric acid reactive substances assay. Ayan. So when you take Fisheries 155, you'll be learning more about this test, no? So, ayan. So the initiation stage, i-ano ko lang briefly, describe ko lang briefly. Fatty acid radical is produced, and most notable initiators in living cells are the reactive oxygen, so which combines with the hydrogen atom to make water and fatty acid radical. So ito yung radicals na naproproduce. And yan yung pwede mong i-read using the PV test. Okay, so the fatty acid radical, sila, is not a very stable molecule. So it reacts readily with molecular oxygen, thereby creating a peroxyl fatty acid radical. So yan yung mga, this radical is also an unstable species that reacts with another peroxide. Okay, so this cycle continues as the new fatty acid radical reacts with the same way hanggang dito. So paulit-ulit sila, and then until ma-reach na yung termination stage. So the secondary oxidation products, no, are the ones that are capable of giving off flavors and odors sa product, no? Yun yung mga ketones, aldehydes, no? Yun yung tinetest mo using T-bars assay. Ayan. So the peroxide test is based on the ability of the peroxide to liberate iodine, no? And the concentration of peroxides is indicative of oxidation during the early stages of lipid deterioration. So, ayan. And it is expressed as milliequivalents of peroxides per kilogram of sample. So, the process there is titration. So, I know you are also familiar with titration na. Ayan. So, T-bars, again, is called, T-bars is an acronym for thiobarbituric acid reactive substances assay. Ito yung assay. Are formed as byproducts of lipid peroxidation. So, ito yung mga byproducts, no, of lipid peroxidation. So, kaya siya tinatawag na thiobarbituric acid is because you are using a thiobarbituric acid as a reagent in this assay. So the off-flavor odor development in lipid and lipid-containing foods is commonly attributed to the byproducts of lipid peroxidation, as I've mentioned earlier. And fats and oils and other lipids reacts with oxygen to form the peroxide. So ito yung mga hydroperoxides. Ayan, and which further decompose to give aldehydes, including MDA, malonaldehyde, and hexenal, and both of which are associated with deterioration in meats and fish. So factors affecting the formation of lipid peroxidation products include how the product was stored, cooked, processed, and if whatever, kung ano man yung chemicals that have been added or preservatives that have been added sa ating products. So pag nag-add ka ng antioxidant or ascorbic acid, no, you can actually retard or reduce the oxidation of your product, no? Okay. So the principle is based on the reaction between 2 moles of TBA and 1 mole of MDA or malonaldehyde, forming the TBA-MDA pink complex. So ang method mo dito that you can use is actually spectrophotometry and HPLC. So yun, pinag-usapan na natin yun kanina. And now, let's go to histamine. So what do you know about histamine? No, this is produced from the decarboxylation of amino acid histidine. So we know that histidine content is related to the species of fish, and not all species are capable of producing histamine, right? Because histamine is only prevalent in those that have relatively high amounts of histidine, amino acid histidine. Okay. Other biogenic amines or amines that can actually manifest itchiness in the products are the following. So, pag mataas yung ornithine, lysine, arginine, no? sa products, it might possible or has potential to produce the protracin, cadaverine, bromidine, and spermine, respectively. So ito yon. And the enzyme that's capable in the formation of histamine is actually the histidine decarboxylase. So ito yung chemical structure of histamine. Yan. So, the histamine-producing bacteria are the Enterobacteriaceae, no? Sila yung nagko-convert or synthesize ng enzyme, no? That's capable of formation of histamine formation. Okay? So, ito yung mga strong histamine producers na mga microorganisms. So, if these microorganisms are high in number sa inyong product, so most likely, and mataas yung histidine nung raw material, most likely mataas din yung tendency na magpo-form siya ng mataas na level ng histamine. Ayan, so this is the threshold, histamine threshold of certain products in some markets. So for Codex or mga regulation bodies. So for Codex Alimentarius, 400 milligrams per kilogram of sample ng histamine is limit. Yan yung limit for fish sauce. For all foods sa FDA, should be 50 milligrams per kilogram. For marine products, it should be 100 milligrams per kilogram. And for fish and other marine products, should be 200 milligrams per kilogram. Sa EU ata ito, 100. Sa US and other parts of the world, siguro nasa 200. Medyo stricto kasi yung EU. No? Why is it? If you might ask, no? Bakit mataas masyado pag sa fish sauce? It is expected kasi na pag fish sauce, pina-age mo yung sauce. So most likely talagang tataas yung level ng histamine, no? But for other food products, mababa lang siya. Kasi dito naman, tolerable kasi mataas naman initially talaga yung histidine content ng marine products. Yes. So for US, ayan, per gram kasi ito, per 100 gram. Ayan. So yan yung limit natin. Ayan, so this slide simply gives you the information about histamine, no? It is a result of time and temperature abuse of certain species of fish. No, and take note, no, histamine formation can lead to this, can cause an illness and can give you these symptoms. All right? And take note na kapag present, once na-form na yung histamine, it can no longer be eliminated kahit pa iluto mo yung product. Okay? So, prevention is better than cure sa case ng histamine, no? That you always have to keep your fish in cold condition or avoid time and temperature abuse sa inyong raw material and sa product. Okay? So, for histamine analysis, you can use fluorometric detection or HPLC, no? And then the principle is based on the amount of histamine in the extract that has reacted with a substance, no? Yung sa limit, ito yung pinag-usapan natin last time that 9 samples must be taken from each batch and must fulfill the following requirements. Mean value must not exceed 100 ppm. 2 samples may have value more than 100 but should be less than 200 ppm. No sample may have a value exceeding 200 ppm. So, ganun. Ito yung scheme, no? In histamine analysis. So, histamine is extracted from the sample with using methanol, and the inferring compounds are removed by anion exchange chromatography. So, mag-extract ka muna ng histamine, and then you do chromatography to remove the to remove the contaminants. Ayan, and then you do a derivatization and detection. Okay, why do you have to do derivatization? This one is a technique used in chemistry which transforms a chemical compound into a product. The reaction derivative with similar structure or called the derivative. You want to get you want to produce a derivative of that product. Bakit mo kailangan gawin yun? Kasi yung derivative na yon ang madedetect ng process mo, hindi yung pinaka-product mismo. Okay, so after that, you detect and you prepare your standard curve, and that standard curve will be used in the calculation of your histamine or histamine content of your product. Ayan. Next is about the TMA, DMA, and TVBN determination. So these are the index, no, that you're going to determine or to test, and then you use the following method. So you use spectrophotometry for TMA and gas chromatography, and you can actually use the same, no, for DMA or dimethylamine detection. And for TVBN, TVBN stands for total volatile base nitrogen. Okay, so you can use Conway microdiffusion method or gas chromatography. Okay, so this one shows the Conway microdiffusion method. Ayan. So we are doing this in the lab. Medyo hindi siya ganun ka-high-tech kasi you will be doing titration afterwards. And from there, you will be able to determine kung ilan yung amount of TMAO in your product, okay, or TMAN. So you will be using this Conway apparatus, no? So it has an inner ring here, and then it has an outer ring. So first you have to prepare your sample, you mean centrifuge, you filter, ayan. And then saan ba dito yung okay, and then you add your boric acid with indicator and sample solution saturated K2 potassium carbonate. All right? So I think yung sample mo is andito sa labas. And then you put your boric acid inside, no? With your indicator. Diyan siya sa loob. And then what do you do, no? Kasi volatiles itong tinetest mo. So you give time, kasi meron yan, no? 60 minutes. You allow it to diffuse for 60 minutes. So all the volatile samples in your, or all the volatile compounds in your sample, no, will be given time to diffuse. So meron kang cover na ilalagay kasi volatiles nga siya. Pag hindi mo siya kinover, tendency lalabas, di ba? So kailangan mo pa rin siyang i-capture. Yung boric acid mo inside, in the inner ring, will capture all the volatile compounds na nasa sample mo. Now, dahil covered ito, nandyan siya sa loob, itatitrate mo ngayon itong boric acid mo. Now, for you to be able to determine the amount of volatile bases na nasa sample mo. Okay? So again, yung principle is you put your sample on the outer ring, you put your boric acid with indicator inside. Okay? All your volatile component, all the volatile compounds present in your sample will diffuse into the boric acid dito sa inner ring kasi covered siya dapat para hindi siya makalabas. Hindi siya maka-escape. Now you titrate. After 60 minutes, you titrate your boric acid, and that's the time you'll be able to determine and calculate the amount of volatile bases na nasa sample. Ayun yung mag-move. Alright, so this one is about indole, no? Indole is produced by bacterial action on tryptophan. Kung yung histamine is from amino acid, histidine, ito naman yung indole is from tryptophan, which is a naturally occurring amino acid in crustaceans and also a putrefactive compound, no? And it is used as an indicator of quality and extent of bacterial activity, no? Especially yung mga indole formers. Ayan. So studies have shown the correlation between indole concentration and bitterness in the taste of your product. So ayan, from a tryptophan, no, in the presence of a tryptophanase enzyme, indole will be produced. Ayan. So ito yung standards, no, quality standards for indole. Ayan. So, for class A, ito siya. Class 2, class 1, 2, 3. Ayan. And ito yung subsampling na ginagawa para kung meron kang 1 to 20 lots, you just take 6 subsamples, 21 to 100. And pag mas madami, 18. Para yun lang sa histamine. No? So for indole analysis, this schematic diagram shows how you do indole analysis. So first you extract the indole by steam distillation with alcohol. And then you'll be able to take the nonpolar compounds. Ito yon sa chloroform na layer. And this includes your indole. Ito, wala na to. Discard nito. And then you make your chloroform layer react with your p-dimethylamine benzaldehyde, and then you also prepare a curve, standard curve, and then you calculate the indole. Now, class, look at this one, no? This is the schematic diagram for indole analysis. Now, when you write your thesis or when you do experiments, no, it is advisable that you also make a schematic diagram of your methodology. So, parang ganito yung itsura niya. Ayan, ganyan siya. So, by just looking at this diagram, one would already see, no, at a glance kung ano yung ginagawa mo sa study mo. So, ayan, ito ay isang schematic diagram. This is just an example of one of the many diagrams that you will be making pag nagtithesis na kayo. Ayan. Okay, last slide na pala siya. So, at this time, no more codes for analytical methods. I also ran out of thoughts, no? Just like our reagents. Kasi pag face-to-face tayo, talagang mararound out tayo ng reagents. Lagi tayong nauubusan ng reagents. Ayan, problema yan sa face-to-face. Okay, any question, class? Any question? Before we totally end. So you watch these videos, ha? May tanong ba? Saan ko ba ito ilalagay? Any question?
None, ma'am.
Okay, let me check your attendance. Ayan, 4 na lang pala kayo. So thank you for staying, no? Up to this time, kahit nakailang cut yung ating Zoom, no? Thank you for bearing with me and see you again next week. All right? Please, by the way, please read ahead your module 5 and 6. So module 6 is the last module that we have for Fisheries 160. For your output in module 4, gumawa na ba kayo ng output?
Ang
Wag mo
mga
kayo-
activities, ma'am.
Ha?
Ang mga activities, ma'am.
Actually, yung isa kasi parang group discussion siya. So we can't do that, no? Kasi hindi naman lahat nagjo-join sa Zoom meeting. So hindi na lang natin gagawin yung output na yun. Tinitingnan ko dito. This one is for, module 6 pala to. Kung makita yung module 4 eh. So, module 4 na study guide, tingnan natin ano yung output. Yung table lang na i-complete. Okay. Kung wala pang nakagawa ng output sa module 4, wag niyo nang gawin. Ipapabonus ko na yung module 4. Kasi you prepare your table na lang for your purpose. Kasi mas magandang gumawa kayo ng table para gamitin yung pag nag-study kayo. Mas madali kasi siyang tingnan na meron kayong. Ito kasi yung natin. Teka. Paano ba ako makapag-share screen ba ako? Ito siya. Ito yung ating output, supposedly. Anyway, party, diniscuss ko na naman yung Teka. Ano yung nakikita niyo sa screen?
Loading pa, ma'am.
Nakikita nyo na yung yung
Okay na, ma'am.
PDF file?
Yes, ma'am.
Okay, ito yung output nyo actually. You have to describe the protein degradation pathway. Tama ba to? Teka, tingnan ko nga. Baka ibang subject to. So, dami nang naka-open. Hay nako, teka. Yung faces natin naka-block sa file title. Ano ba yan? Hindi siya ma-remove. Ayan, fish 160. Ayan, module 4. So, partly na-discuss na to. Ito, gawin nyo tong table for your purpose na. No need to submit. Okay? And ito kasing group discussion, ideal to if all of you are attending the Zoom meeting. Kaso you don't. Bihira lang yung uma-attend. So, sige, pa-bonus ko na to. Pero tinitake note ko yung mga nag-attend ng klase. In this one naman, sige, ano na lang, mag-a-announce na lang ako kapag kailangan nyo pang gawin ang sa module 4, module 5 and 6. So, for the record, bonus na yung module 1, ay module 4 na output. Okay? So, that's all for today. Please study ahead yung module 5 and 6. Siguro hindi na ako magpa-per slide na discussion sa 5 and 6 kasi it will be a total of 6 PowerPoint lectures. So, lang natin, tingnan natin yung mga kailangan lang i-elaborate. Skip natin yung iba. Would that be fine?
Yes, ma'am.
Yes,
ma'am.
Sige. So, that's all for today. Thank you. Happy weekend. Bye.
Thank you,
Enjoy
ma'am.
your time. Rest.
Thank you po.
And be healthy. Bye-bye.